I'd like to thank the organizers for allowing me to talk. I've really enjoyed the meeting. And I'm going to talk about comparisons of different computational methods to predict the impact of regulatory variance. So we know that there are thousands of non-coding SNPs from GWAS that are associated with common human disease. And most of these fall in active regulatory regions by various assays. And it's easy to dismiss these, but I think there's a lot of biology of human disease in trying to understand these. So my lab has been focused on computational techniques to understand regulatory variation and its impact on human disease. So our most successful approach has been to train SVM-based classifiers using K-mer features. We train these models on functional genomic data sets. And this identifies a regulatory vocabulary, or the set of active transcription factor binding sites that are determining the chromatin accessibility or transcription factor binding in that cell type. And we've showed in a recent paper that this method can predict the quantitative impact of the mutation with several comparisons to experimental data. And I'm going to show in this talk that it's significantly more accurate than alternative approaches, either PWM-based approaches or deep neural networks. So let's start with a clear example of the impact of a regulatory variation on a human phenotype. So this is a SNP that's highly associated with an increase in LDL cholesterol levels and an increased rate of myocardial infarction. It's located in this region of 6 genes. There are many SNPs in this locus, and many SNPs have D prime, which is large, which means they're transmitted along with the associated SNP at a very high frequency. So it's really indistinguishable from all these other SNPs are indistinguishable from the main associated SNP from the GWAS study. But Dan Rader's lab further explored this locus experimentally and found that this particular SNP is associated with about a 4-fold change in the expression of 2 neighboring genes. And I don't want to give anyone a heart attack right now, but 5% of you are homozygous for the bad allele, right? And about 35% of you are kind of in bad shape. So they were able to narrow down on this region more closely, and experimentally tested each one of these SNPs that are transmitted along with the associated allele 646776. They tested each one individually and showed that just one of these really reduces the expression back to the major allele from the minor allele. And the computational method I'm going to describe, with that method we scored all these alleles and we only get one of them getting a significant score. And it actually is the one that they validated experimentally, and it's in the DHS regulatory region, and this is the actual GWAS-associated allele. So this is another example of one of the 5,000 that has yet to be validated, and this is a SNP associated with type 1 diabetes. When we train our model on a cell type that's specific for this autoimmune disorder, Th1 cells, we get the regulatory vocabulary. We can score each of these SNPs that are in LD with the associated allele, and we find just one gets a large score, and it disrupts IRF1/2 binding site. So the basic computational model we proposed is to define a set of cell type specific enhancers, either from DHS, from ENCODE, or ATAC-seq also works really well. And then we train our gapped K-mer-based classifier to learn the set of binding sites, which we call the regulatory vocabulary, appropriate for that cell type. And then we can calculate how each SNP changes that SVM score, right? So we take a set of positive regions, enhancers, or open chromatin regions. We take a set of closed regions or non-active regions in the genome. We select the negative set to be GC matched. We train a classifier which finds this boundary, and then we ask any SNP, how much does it change your position relative to that boundary? The way we build this classifier to resolve longer binding sites is the really complicated part of this problem. So we found that K-mers work up to about 8, and then their accuracy starts rolling over. In order to get a robust set of features, we had to use gapped K-mer counts, and we developed a fast way to evaluate this kernel. So this is in a series of recent papers, and it builds on earlier work by Leslie and Noble. So once we have that set of K-mer weights, we can score any particular mutation, like this C to T mutation, by the way it changes the weight of all the flanking 10 K-mers, right? So this 10-mer to the left, that C changes to a T, that weight changes from that to that, right? Here's the big effect where an NF kappa B binding site is mutated from that C to a T. This weight goes from 5.5 to 1.7, right? So it goes from a strong binding site to kind of a moderate positive binding site, and the sum of all these changes gives us the score. So there's one really great data set where we can use to evaluate this model. It's a set of DSQTLs published in Nature by Jonathan Pritchard's group. So this is a set of SNPs whose genotype affects the DHS sensitivity at that locus. Right? So the CC is very accessible, TT is inaccessible. So this was done on a set of 70 individuals in lymphoblasts, so the genotype varies across that set of individuals. When we compare our predicted score to their effect size beta, which is the slope of that line, we get good agreement, a good correlation between our prediction and their measurement. But this is an easy test, right? Because it's only scoring kind of basically the sign of the change. And a much harder test is to pull out those set of DSQTLs that are actually changing the chromatin state from a negative set that's much larger. That's kind of the problem we're faced in interpreting GWAS data. So every GWAS-associated locus has about 50 SNPs in LD with it. And so that's the negative set that we should use as a way to pull out which of the DSQTLs in that locus has the causal effect. So if we train on the appropriate cell type and then ask, does every positive DSQTL in Jonathan's set get a larger score than any region in this, any SNP in this negative set, we can calculate the precision of that prediction. And you can see that the gapped K-mer method using 10 mers is much more accurate than using just 6 mers, which was our previous model, or these two previously published methods to detect causal SNPs or conservation. So they have a very low kind of predictive accuracy on this test. We also directly tested this model experimentally by making mutations in 2 melanocyte enhancers and directly comparing delta SVM score with the change in luciferase activity, and we get a high correlation. And we also got a high correlation with 3 previously published massively parallel data sets. So ultimately, we want to predict the effect of a SNP on enhancer activity. There's a complication with predicting enhancer activity using the DHS data that we have to train on, and that is that while not most, but some fraction of the DSQTLs are also eQTLs, which means they have an impact on enhancer activity or the expression of some gene. In their native context, about 1/3 of the DSQTLs are actually repressive. So we've shown that delta SVM correlates positively with DSQTL beta for all the DSQTLs that are also eQTLs. But about 1/3 of the eQTLs are anti-correlated with the DSQTL beta, which means that the region becomes more open, but the gene that's associated with that region goes down in expression, right? So we only get a positive correlation between delta SVM and eQTL beta if we separately consider those 2 classes. So instead of testing different methods on expression, I'm going to use DSQTL as the test case. So the gapped K-mer SVM model is really totally encapsulated in the set of weights. And here I'm showing the distribution of all of these weights. There are 500,000 of them, 500,000 10 mers. And the distribution has a long positive tail and a smaller negative tail, and there's a lot of stuff in the middle that doesn't matter, right? So this is the very top of the distribution of weights. You can see CTS CTCF binding sites. This is an NF-kappa B binding site that I showed before from Jonathan's paper. And within kind of this intermediate range of the tail are tenomers that match all of these PWMs. All right, so the weights that we pick up in the gap-Kamer classifier kind of encode binding sites for a large set of cofactors that can be recognized from PWMs. And that's where the accuracy is coming from. So we've shown that the gap-Kamer SVM AUC is higher than the best single PWM on the full set of ENCODE ChIP-seq data. But we can build a classifier that uses PWMs as features, right? So we've been showing that these high-weight Kamers look like PWMs that we can recognize as cofactors in lymphoblasts. So why don't we just build an SVM using PWMs? So we can do that. We just score all the sequences in the positive set and the negative set, and we use those PWM scores as features in an SVM. We've actually done that a long time ago. And we can show that the accuracy of that classifier in purple is really quite good, right? But then when we go to test that classifier on mutations, so we now score both alleles, the reference and the alternate allele, at a DSQTL. We calculate all the changes in the PWM scores. We add them up to give a change in the SVM score. Now we get a rather low precision of the prediction for DSQTLs. All right. So compared to the gap-Kamer method, we get a pretty low accuracy or low precision. Another way we could do this is to use a previously published method to score the affinity change for all the PWMs in one of these databases, and that's shown in green. All right. So this is asking, does every mutation in the positive set give a larger score than every mutation in the negative set? And that's a very bad classifier for Jonathan's DSQTLs. But if instead we just look at the affinity change for the top 20 PWMs that get large weights in this classifier, we get a result that's comparable to the full PWM SVM. So Kamers work better than PWMs. And it could be that these databases are incomplete. It could be, as a lot of people talked about in this meeting, that improved PWM models would help. I think that the main problem with PWMs is they do well at classifying the strong binding sites and not so well at the intermediate binding sites. We've also compared to the seqGL method, which was recently published. We get kind of a systematically higher AUC on the test set than seqGL, but when trained on DHS, they get also a reasonably good test set AUC, but when scored against the DSQTL set, the accuracy of seqGL is lower and a little bit lower than just using 8 MERS in a Kamer SVM. So the last comparison I want to make is to Deepsea, which is a method that was a deep neural network method that was recently published. In that model, they have 3 large convolution layers and 3 pooling layers. And we took that model, which was originally trained on the full set of ENCODE data, so a full collection of 918 different data sets. And we were concerned that training on all the data would bias your accuracy toward overrepresented cell types and transcription factors in the ChIP-seq data set. So we retrained that model on a single cell type. And this required a substantial reoptimization of the kind of parameters of the network. But when we did that, the AUC was a little bit better than GKM SVM. And kind of we've known and reported previously that using a longer word is better when you have a large training data set. So we did that, and then we get comparable accuracy to deep neural networks. So the recently published deep neural network method is comparable in accuracy on the DSQTLs when trained on GM12878 DHS. So I want to say that training on the larger data set always improves our accuracy. So if you compare to our method, I suggest that you grab this new implementation that can handle very, very large training data sets that Dongwon just released. And the final point I want to make is that the prediction accuracy is always dependent on training on the right cell type, right? So we only get a high correlation between any of these data sets when we train our model to detect the cell-specific vocabulary that's appropriate for the assay. And especially when we compare to validated causal SNPs for LDL cholesterol, which I just showed, the flanking non-causal SNPs are in gray, and the validated causal SNPs is the delta SVM score for the validated SNP. And for a prostate cancer-associated allele, we also only pull out the validated SNP if we train on the appropriate cell type, so the prostate cancer cells from ENCODE. And in the full ENCODE data set, there are really only 2 representatives of this cell type. And so we really are concerned that training on the full ENCODE data all at once would not pick up this effect. And I'd just like to thank Felix, who's here, who did a lot of the comparisons with CGL and STRAP, and Dongwan, who did all of the deep neural network comparisons and reoptimization of that code. And I want to thank Anshul for useful discussions about deep neural networks. And just an ad, I have postdocs available, and I'm in the Department of Biomedical Engineering at Johns Hopkins, which is the home for our computational program. So thank you very much.
We have time for one quick question.
I guess I was just wondering, as far as you noticed such better improvement with gapped k-mers over PWMs, is there any relationship between like which methods were used? Did you look at all at which like in vitro methods were used for creating the PWMs, or which algorithms were used to get PWMs out of the in vitro methods?
So I've played with the set a little bit. Like we've generated, I can either use the full like transfag JASPAR, I can filter by year and kind of use the more recent stuff. I don't really, this always happens, right? So no matter what set I look at, using a large set of PWMs will learn features that can successfully train on any single cell type, right? So as long as I've got an SVM with a complete representation of the features, and somehow I'll get good accuracy on the test set, right? Then when I go to evaluate the impact of variance, it always drops.
Do
Right.
you think this is related to that PWMs can't capture or that these PWMs don't capture the sort of like lower affinity binding sites accurately?
That's what I think. That's my guess. But it could also be that there are features in the k-mer weights that describe kind of deformation properties of the DNA, or some like histone kind of affinity that are somehow folded into all the k-mers in combination with the transcription factor binding sites. And then perturbations of certain binding sites will kind of strengthen the histone affinity. Other perturbations to the binding site will kind of weaken it, and there's a competition that's going on that's reflected in this full list of 500,000 numbers, right? So it's all folded into here, and pulling it apart, if you want to do that, is not trivial.
Yeah. Thank you.
Thanks.